Morning, folks. My name is Cara Schreiber, and I am the lab supervisor for the 1802 lab. Today, I wanted to go through the virtual lab exercise for exercise 3 seedless plants. So we'll go over the specimen and some microscope slides for liverworts, mosses, and ferns, and then we'll also look at just a couple of specimens for club mosses. And this is in lieu of the normal virtual lab that we have, but since Monday is Memorial Day, I want you to take that day to have some time to yourself. And so you can do this anytime between Sunday and when your worksheet, exercise 3 worksheet is due on Tuesday. So just watch this video. You can pause it between the different specimens. So if you get through liverworts and you want to take a break, just pause the video and come back to it when you're ready to do mosses, or you can do it straight through. So I'll also be switching between different spotlight views and different camera views. So please just be patient with me as I navigate the technical aspect, but I look forward to going through seedless plants. They're great, and let's go ahead and get started. I'm going to remove that spotlight and go ahead and spotlight over here. All right. So for liverworts, I wanted to start by showing the part of a liverwort that we normally see out in the environment, and so that's going to be this sample here. So let's zoom out just a little bit. So in this sample, you can see a liverwort, and right now I want to just talk about what it looks like so that we can kind of remember this. So it's green, it's leafy, it's called a liverwort because it's liver shaped, so it kind of has these lobes. And this is going to be our gametophyte generation. I remember that because the gametophyte generation is dark leafy green, and it also is generally very fragile. And so we can see here that this is a very thin tissue type. Let me get a probe here, and then I can show you the rhizoids underneath it. So this is a really thin tissue, and if we lift it up, we can actually see underneath here are the rhizoids. Let's see if I can find a really good sample here. So here you can see some fibrous material, and so that's going to be the rhizoid. Zoom in so you can really see it here. So these fibrous parts, those are the rhizoids, and rhizoids are what connect this thallus, this gametophyte generation of the liverwort thallus, to the substrate. And so here it's living in soil. It's pretty common for liverworts. And the important thing to note about these rhizoids is that although they're anchoring the thallus to the substrate, they are not actually procuring water, so they cannot absorb water. They're just for anchoring, they're just for support. And so liverworts are a non-vascular plant. It's a plant that cannot obtain its own water through vascular tissues because it doesn't have vascular tissues. It hasn't happened yet in the evolution of plants. And so this organism has to stay really low to the ground and stay really moist. And you can see the sample that I brought in. I put some water in here, and this sample actually came from right next to a stream bed, right? So it needs to stay really moist. When we think about our gametophyte generation, I want to put it in the context of the life cycle. So I'm going to accompany these specimens with some drawings as we go through. Start here. Let me grab my supplies. So with our liverwort, the gametophyte generation, I'm going to do a little caricature. And so when I think about the liverwort gametophyte generation, I kind of draw like a pond shape. Okay, and so that's going to be the thallus, right? The thallus of our gametophyte generation. And for gametophyte generation, I'm just going to put a big old G, okay? And so this particular thallus does not have any reproductive structures on it. I just found this one, and sometimes they have reproductive structures on them, and sometimes they don't. So this particular sample does not. But if I was looking for the reproductive structures, what would have to happen is that this thallus, this gametophyte generation, would need to be at a point where it is sexually mature. So Where does the gametophyte generation come from? Well, when a spore lands and germinates on the ground, it's going to germinate here, and then it's going to start putting out this prothallus, or just like a little chain of cells. And as it grows, this chain of cells is going to get larger and larger, right? And this whole process is happening through mitosis. Eventually, this chain of cells is going to turn into our thallus. And so at this point here, immature, not able to produce reproductive structures. At this point, once the thallus is formed and it's matured, if it's sexually mature, it will be ready to make sexual reproductive structures. And so for our liverwort, what are the sexual reproductive structures? I'll give you just a second to think about it while I edit my picture here and turn it on its side. All right, so the sexual reproductive structures that the liverwort thallus is going to make is going to be, I call this our palm tree, okay, the palm tree structure. And then we also are going to have an umbrella structure, okay? So the palm tree structure, you just have to memorize this, is going to be the female reproductive structure, and that is the archegonia 4. And the male structure here kind of looks like an umbrella. That's going to be the antheridia 4, all right? And this is going to be a theme for us as we go through the seedless plants. Arca, okay, arca is going to be for female, and anther is going to be for male. And I remember this because I know that anthers on flowers are the male part, and arca is, and you just, I mean, on some levels you just have to memorize it, and that's okay. But so we have the archegonia 4, and 4 is, the archegonia 4 is talking about this entire structure in antheridia 4. And then this is the antheridial head, right? Just the head part. And this is the archegonial head, just the head part. Okay, so why are we talking about male and female? Well, these are reproductive structures. And so these reproductive structures are going to produce gametes, all right? And so we're remembering that our green leafy gametophyte stage is going to produce gametes, and gametes are going, we're talking about here sperm and egg, right? So luckily plants use the same system as we do. So luckily the female is going to produce an egg and the male is going to produce sperm. So that's easy enough to remember. And so the males in this structure are going to produce these sacs. And inside the sacs, they'll produce the sperm. For the females, they're going to produce these vase-shaped structures with a egg in them, all right? And I want to show you an actual diagram that shows a microscope slide, because it's a little bit easier to see there, but I like drawing this caricature as we go through so that you can kind of imagine what's going on. I have these really nice old lab books here, and they've got great pictures and diagrams, all right? So here we have our leafy gametophyte liverwort, and then here we have some reproductive structures. They're palm tree shaped, so these are going to be the female archegonia 4. And the archegonial head is going to look like this if we did a longitudinal section. Let's just zoom in here. And when we zoom in, when I said that they're going to produce these vase-shaped structures, this is what I'm talking about. So here's a palm frond, here's a palm frond, and underneath the palm frond, you can see these vase-shaped structures. And in that pink dot right there in the middle, that's where the egg is going to be produced. So this is the archegonial head of the liverwort. And these right here are being produced in the archegonium. This structure here, the base-shaped structure I was talking about, is the archegonium. Archa again, so archa, A-R-C-H-E, is going to be a theme. So this is the archegonia 4 with the archegonial head and the archegonia that produces the egg. And then if we move down here, this is what the antheridial head looks like, and it's kind of turned on its side, so I can turn it in a way where you can see it straightways. But here are those sacs that I was talking about, and these sacs are called the antheridium. So again, here's a theme: the antheridia 4 is the whole thing. The antheridial head, they're going to produce antheridia, and the antheridia are going to be what produce the sperm. So in pink here, this is where the sperm are being produced. All right, so if we come back here to our sample and we're looking at our thallus, and the thallus is sexually mature, and we are drawing a caricature here of what that looks like. We have our archegonia 4, our antheridia 4. The archegonia 4 is producing archegonia with eggs, and the antheridial head is producing antheridia, which produce the sperm. And so just like in all forms of life here, the sperm are going to be released and meet the egg. And because this organism requires large quantities of water, the sperm, well, actually, it's not because it requires large quantities of water. The sperm are the reason that this organism requires large quantities of water, and the fact that it doesn't have any vascular tissue. But the sperm is pretty important here too. So the sperm requires a large quantity of water in order to swim to the egg. So once the sperm is released, it swims over to the egg, and the egg is fertilized. So then we're going to have, let me get a different pen. So then we have our palm tree structure, all right, and we have our base shape and the egg has now been fertilized. So this is now a zygote. And let's just really quick come back here. So we know that the gametophyte generation is 1N. 1N, gametophyte generation is 1N, that's the ploidy. And so the structures that this gametophyte makes will also be 1N, right? Because they're being made through mitosis. Therefore, the products that this structure make are also 1N because we're still undergoing only mitosis. So this is different than in humans. Mitosis is how the gametes are made from the gametophyte generation. So all of this is 1N. But once the sperm, which is 1N, fertilizes the egg, which is also 1N, we produce this zygote. And I wanted to bring this up because now the zygote is going to be 2N, right? So fertilization of a 1N and a 1N is going to produce 2N. So we now have a 2N structure here, the zygote, and the zygote is going to start growing to produce the sporophyte. And the sporophyte is going to kind of bulge out from the bottom of this palm frond. So I'm just going to adjust my picture because we're going to show a time lapse here. So it starts bulging out and then it bulges more. And eventually this whole structure becomes pretty large. And this is the sporophyte, right? And the sporophyte is 2N because it came from that 2N zygote. Zygote's now gone because it's matured into the sporophyte. So I'm going to put a big S for sporophyte generation. And as this balloons out, the sporophyte produces spores, sporophyte spores. And the sporophyte produces spores by the process of meiosis. So meiosis is the process that takes a 2N structure and produces 1N structures, all right? And so in this particular case, the 2N sporophyte is going to undergo meiosis to produce 1N spores. And once the sporophyte is filled with lots of spores and it bulges out a lot, it's going to burst. Okay, this is going to be my version of bursting. and the spores are going to be released out into the environment. There's still one in. So the sporophyte undergoes meiosis to produce spores. The spores are released from the sporophyte generation, which is still attached to this archegonia 4 of our gametophyte generation, which is attached to the thallus of the gametophyte generation. And once these spores are released into the environment, they land and they start to grow into the gametophyte generation. So that's our life cycle. And we come right back here to the gametophyte generation, which is the thallus. I also wanted to show you real quick, here are some dried samples. And I think it's really important when we're in lab to acknowledge the dried samples and how they look. So they're not going to be as brightly colored, if that makes sense. But let's take a look if we can identify some of the same really important structures of our thallus. So here we can see the thallus. It's still lobe shaped, and you can still kind of see it has these parts that kind of extend out to the side. It's still really flat and close to the ground, indicating that it has a high water requirement. And then here's a little piece of the thallus that I took off, and you can see these structures here extending from the side. So those are the rhizoids here. So this yellow structure is just kind of a dried out, discolored version of the thallus here. And then we have these rhizoids. And so we can do a compare and contrast. Let's see if I can pull off a little section here. So here's our live specimen, and you can see here the rhizoids are extending out. You can get in pretty close with this camera here. Yeah, look at that. It's a beautiful picture, right? So thallus, rhizoids, both part of the gametophyte generation. We don't have a live sample for our sporophyte generation. I can, however, show you this diagram, and I think this is a really great depiction of what's going on here. So if we were going back and talking about the development of the sporophyte generation, here is a longitudinal section or a cross section of the archegonial head. All right, the archegonial head here, and so the archegonia would have been here. So just imagine, we'll start with stage 1. So at stage 1, we're looking at this base-shaped structure, which is the archegonia, which originally held the egg, but at this point, it's been fertilized and it's starting to produce a sporophyte generation. So here's stage 1, and stage 2, it's starting to balloon out a little bit more. Stage 3, it's ballooning out even more, and then finally, it's ballooned out completely. And then this bottom, so this is the whole sporophyte, and it's kind of ballooning out from underneath those palm fronds of the archegonial head. And then right here, this is the spot in which those spores are produced. All right, so stage 6, mature, and it's ready for the spores to be released. All right, liverworts. Not very familiar to most of us, but a really interesting organism. And in Boone, you can find liverworts near creeks and ponds and in moist areas pretty readily. So the next time you're out hiking, definitely take a look and see if you can find some of those. All right, and so evolutionarily, when we're talking about liverworts, this is kind of our first group after our ancestral algae moved from water to land. And so that's a really important step. And as the algae evolved to be able to live on land, the first kind of base step that we talk about is liverworts. So liverworts very close to the ground, really requiring a lot of water. And then we're moving from there to mosses. And so mosses are one step up. They're able to live just slightly further away from water. They have a smaller or less water requirements, but still we think about mosses being in really moist areas. So we know that they still have some moisture requirements. So I wanted to show a couple of different moss samples here, and these are all dry, but I think they're still in pretty good shape here. So let's take a look. We've got this one that's kind of like a mat. So this moss, and I'm trying to show you samples that you're familiar with. So mosses, the generation that we're familiar with is this one. And this is the gametophyte generation. So here's a mat of moss, all right? And then we've got this mat of moss that's a little bit lower to the ground. And this one's like really kind of a little bit taller, maybe an inch tall and fluffy. And this one's a little bit more mat-like. This one's pretty mat-like as well. Or like, you know, just growing closer to the ground. And then this one is actually growing on a piece of bark here. So lots of different versions, and all of these are dried. So when we look at dried samples, we know that they're dried samples because they're not verdant. They're not really bright green, right? So these are dried samples, but still important to take a look at here. And when we think about the moss, we normally think about it in this whole mat, but actually each moss specimen is just this little stem-like thing. Zoom in on this one here. And so it's just the stem-like structure with these fuzzy bits that would normally be what you would think of as the like little leaflets. So I've got a live sample, and I'm going to take a little sprig off of it as well so we can compare. All right, so here's the live sample. All right, so here are moss specimens. So 1 specimen, 2 specimen, 3 specimen, dried, live. All right, and so let's go back to our drawing board and talk a little bit about where we're at here. So zoom out here. So here's our moss sprig, and kind of place them so that you can still see them. So moss, when we think of a moss, we're thinking about these moss mats, and then as individuals as these sprigs. So I'm going to draw a couple of sprigs, and your drawing doesn't have to be amazing because this is enough to distinguish it between what we drew earlier, which looked like a pond, right? So that was our liverwort. These are mosses, so they look very distinct, all right? So we have 2 moss specimen, all right? And they are green and low to the ground and kind of fragile. So these are going to be our gametophyte generation. So the gametophyte generation is always fragile green. And so I'm going to have 2 different specimens here because with moss sprigs, they are gendered. So we have a male and female. And it's the same thing as we were talking about with liverwort. So we have a moss spore that lands, and when it germinates, it's going to produce a chain of cell or a prothallus. And so as this is, or protonema, the prothallus is prothallus for when it's about to make a thallus, protonema for when it's going to make a structure that is not a thallus. So, but in both cases, the prothallus or the protonema are both indicating that the spore has germinated and is creating a chain of cells. And this chain of cells will eventually produce, through mitosis, our moss sprig. All right, so that's kind of where we are in the life cycle here. So we have 2 sprigs that both came from that process. One is male, one is female. And when they become sexually mature, they're going to produce a sexual reproductive structure on the top. And honestly, it just looks like more moss fuzz. It's not distinctive the way that we saw in liverworts, where there was that really awesome palm tree structure or the umbrella structure. In this case, it's just a little extra fuzz, okay? And the little extra fuzz is going to be indicative of the archegonial head or the antheridial head. I have to keep switching out these markers here. So arca, again, archegonial head, and antheridial head for the male. All right, so at this point, they are sexually mature and they've produced a sexual reproductive structure. And I want to switch over to these diagrams that we have in the books because they're really great for looking at what these structures look like microscopically. So we're looking at the head groups, right? And so here we have, just ignore this part. So I'm going to just take my hand and cover this up here. So here we have the antheridial head. So this is the top head part of that male moss sprig. And what we can see are these sacs, or the antheridia, that are producing sperm. So we saw this with the liverworts, when we looked at the antheridial head also produced sacs that produce sperm. So that's familiar. And then here, the archegonial head, or the tip of the female moss structure, this one is going to have a different look, actually, visually very distinctive. And if we zoom in even further, hopefully we can find those vase-shaped structures. Yeah, all right, so we had to zoom in a lot. But here's the vase-shaped structure here, and that little pinpoint right there in the middle, that's the egg. All right, so egg, the archegonia is the vase-shaped structure, and then the archegonial head is the entire head of the tip of this moss sprig. So imagine this moss sprig had this special tip on it. All right. So we talked about how these structures are reproductive structures. And so if we zoom in, I'm going to make my sprigs again. My marker's dying again. For the female structure, the archegonial head, the egg is in here. For the male structure, the antheridial head is here. And so the sperm are going to travel, hopefully, to the egg, but it requires water. So this process, again, is still requiring water in order to occur. These organisms are still non-vascular. They do not have vascular tissue, and so therefore they cannot pull water up into their tissues. Any kind of rhizoids that may be present would be on the underside of this patch of moss, and they would serve solely to anchor the moss to the substrate. They cannot get water. They cannot bring water into the plant because it's non-vascular. But this brings up a really great point before we move forward, is that when we're looking at this mat of moss, very spongy, right? And very soft and spongy. And so this soft, spongy nature makes it also very absorbent, like a sponge. So mosses are very, very effective at absorbing water from the environment and then holding them within these moss mats. So that's an advantage of having a moss mat over having the liverwort thallus, which is not going to hold a lot of water inherently, right? Because it's just a flat structure that has a lot of surface area. And when we talk about surface area, we talk about things drying out. So liverworts aren't as capable of holding water for themselves, whereas mosses are a little bit more capable of being sponge-like. But that really helps them specifically for this instance that we're talking about here, where we're looking at the reproductive cycle. The male moss sprig has produced sperm. The sperm are going to be released, swim over to the egg, and then the egg becomes fertilized. Once the egg becomes fertilized, it's no longer referred to as an egg. We refer to it as a zygote because it is now 2N, right? So our gametophyte generation of the moss, we're doing our big G here for gametophyte, is 1N. It's going to produce 1N sperm through the process of mitosis. The sperm is going to swim over to this 1N egg that was also produced by the process of mitosis. And so our female gametophyte is also 1N. All right, so just backtracking there to make sure that we're keeping track of the ploidy here. And once we have all of that and the egg has been fertilized and it's a zygote, it's 2N, and it begins to grow, the zygote begins to grow through the process of mitosis to produce this structure. So at first it's just a little structure like this. Mitosis continues to occur. It grows bigger and it's more like a structure like this with a little cap on it. And then finally, the cap falls off and we're left with this structure. Okay, so here is an immature sporophyte. So we're going to do big S for sporophyte. It's 2N and it's slowly developing. And you'll notice that it's developing out of the archegonial head of this female moss sprig. That's really important. Okay, so sporophytes will only grow out of female moss sprigs because that's where the fertilized egg ends up. So we have it growing out of here, and the sporophyte itself right now it's going to be a little bit green. So just imagine, I know this is a caricature, it's all in black, but just imagine you have your bright green moss sprig, and then the sporophyte's going to be a little bit green. But it doesn't really have to be very photosynthetic because honestly, it's just living straight off of the female moss sprig. So at this point, it becomes brown, and it's not really photosynthesizing whatsoever, and it has this capsule under here, and then it has this little furry cap on top. So I like to think about those cartoons from back in the day where they had the little gnomes running around. This is like a little gnome hat sitting on top. And then once the gnome hat dries off and the capsule underneath it is mature, then the cap falls off and the capsule is left without this little cap here, okay? And so they have technical terms, of course, but I'm trying to talk about it in a way that's a little bit more, is a little easier to understand at first. So I can actually show you this structure on a couple of different moss specimens that I brought in. So let's take a look at some sporophyte. So here I have this container, and down here we have our gametophyte generation. So we have our moss sprigs, and it's pretty moist down here. I'm just going to give you a feel for it. Spongy and moist and fluffy and wonderful, right? Because that's how mosses are. And then if we take some of this out, we'll look at a couple of these sprigs. And what's great about these sprigs is they're in all the different time points of the sporophyte generation. So that's how convenient. So let's take a look. Right. Yeah, look at that. All right. So here, let's start with this one. So here are the beginnings of this sporophyte generation growth. So you can see a little bit green here, and they've got these little tips at the top. These tips are the capsule, are going to be the capsule eventually, but they're not quite the capsule yet. They're still immature. And you can see that they are pointy on the top. That's because the little cap is still intact. And so that cap is called a calyptra, and it's kind of like a tissue-like paper, tissue paper layer like thing that is going to protect the capsule. And actually this calyptra is just leftover tissue from when the sporophyte burst through the archegonial head. So it's just leftover tissue kind of from the archegonial head here. And as we're maturing, these little spiky tips, these spiky tips are going to mature into the capsule itself. So let's take a look here. I can zoom in. This is three-dimensional, so it can be a little bit tricky. But here we can look at a capsule. And let's look at this one. If I can manage this. Let me turn the lights off, and it'll give me a lot more context. All right, that's much better. All right, so here you can see the calyptra is this tissue paper like structure here at the tip. Yeah, let's go like this. So you can see that tissue paper right there on the top. So that's the calyptra. And then if we pull that off, because it's dry right now, it was ready. So there's the little calyptra cap. That's what I was talking about, the little caps. Like little gnomes would have, all right? And then we can look here and we can see our capsule, which is the important part of our sporophyte generation. You can still see that it has a little tip to it, okay? And there's actually, there's a second cap, and this cap is going to be called the operculum. So the operculum is the top to the capsule, and the top to the capsule is going to keep this structure, which is kind of like a barrel-like structure. It's going to keep that structure closed for right now. So the cap is keeping the capsule closed. And that's because, as you can see, this is green. And that means that it's still a little bit immature. It's not quite ready for its final task. So what's its final task? Well, the final task is to produce the spores, right? Because this is the sporophyte generation. And so sporophyte generations produce spores. So let's get back here. So we have our caricature, and I showed you when the little cap fell off. So there's your calyptra. And then in this case, there's still a little cap here that's keeping this capsule closed because it was green and immature. But once it's mature, this 2N capsule is going to undergo meiosis. And once it undergoes meiosis, it's going to form those 1N spores inside the capsule. Once it's mature and the spores are mature and they're ready, that operculum, that second cap, will fall off. So I'm going to put that down here. It's now fallen off, and this is open. And there are actually these little teeth here on the outer part of this opening to the barrel, the capsule, and these are called peristome teeth. And really their purpose is to just fling mature spores out into the environment. All right, so we have our 1N gametophyte. It's reproductively active. The male part produces the sperm, meets the egg is fertilized, zygote is formed. We have our sporophyte, it's immature, it's growing, it's still immature. It loses its first cap, so the calyptra, it continues to mature. The capsule is formed, which is 2N because it's formed from the 2N sporophyte. This capsule is going to undergo meiosis to form spores, sporophyte spores. And once the spores are mature, the second cap, the operculum, is going to fall off, and the peristome teeth like that surround the opening to this barrel-shaped capsule are going to fling spores out into the environment. And then guess what? We come back to our initial stage where the spore lands on the ground, it germinates, and it begins to form this protonema, extending longer and longer until it becomes our moss again. All right, great job. Thanks for sticking with me here. So we just made it through liverworts and mosses. So I'm going to take a quick pause, and then what we'll do is we will come back and talk about club mosses and ferns. All right, folks, back. So we're going to talk a little bit about club mosses. You are not responsible for the club moss life cycle, but I do want to just put in a little plug for club mosses here because they're an incredibly important part of the story of the evolution of plants. So we just talked a little bit about how there's this ancestral protist that was an algae, a green algae, and it was able to survive eventually on land. And so that was the beginning of land plants. We started with some very ancestral organisms that would be similar to a liverwort. So a liverwort is going to have a high moisture requirement. And one thing that we didn't talk about, but is a really important part of this story, is that liverworts are gametophyte generation dominant. And when we say dominant, what we mean by that is that they're both the longer lived and the larger, like physically larger generation of that plant life cycle, right? So plants have two life cycles and they spend a little bit, around half in both, but really ultimately there's no such thing as exactly half. And so what happens is there's a reason why plants shift a little bit away from halfway. So at the beginning, plants are more similar to their ancestral algal, their algal ancestor, excuse me. And so they're very water dependent, and their main generation is very similar to what it was in water, right? So that gametophyte generation, and that gametophyte generation is very fragile and green gametophyte. And that's where they spend the majority of their time. And then they have a little bit of time as the sporophyte generation. Ultimately, plants move further and further away from being gametophyte dominant, because as they're moving onto land, they can't stay gametophyte dominant. That's because the gametophytes are green and fragile, and gametophyte generations are not very well adapted to a terrestrial environment. But as the sporophyte becomes more and more dominant, it develops more structures that allow it to be more terrestrially based or to live on land. So one of the big turning points in plant evolution after liverworts and mosses is the club mosses. So the club mosses are our first vascular plant. So they have some vascular tissues that allow them to take water from the soil and transport it to the tips of their plant tissues. And so this allows plants to get a little bit taller because they don't have to just sit on the ground and absorb moisture. All right, so what is that going to look like? What do club mosses look like? Well, they can have a lot of different really interesting forms here. So one of the forms is very moss-like actually, and they are called club mosses, which is infuriating because it further adds to the confusion. This is not a moss because mosses are non-vascular, and this is a vascular plant. So this is Selaginella. That's just a genus of a common genus of club mosses. And what we can see is that Selaginella, if I tip Selaginella on its side, Selaginella has a little more height than what we saw with the mosses. So mosses were maybe getting up to an inch. This is up to 2 to 3 inches. And actually Selaginella can be a big old puffball here. So it might get even bigger. But Selaginella has vascular tissues. And so what it's able to do is get water from here to the tips using those vascular tissue. So, and we have these support structures here, but ultimately down here in the soil, this Selaginella is going to have root-like structures, roots. So roots that can suck up water and then transport it through the stems of this organism, the specimen, and up to the tip. So the height of that specimen kind of tells us that it has to be vascular, because at that point it's growing too tall to just absorb water from the ground. This is another great example, and we have to zoom out even more here. So here's another club moss, and this club moss has more these really long stem-like structures, and it actually is kind of a leafy thing, so like it leaps to the side, but let's just look at it being long like this, because that height that you're seeing is 100% enabled by the fact that this is a vascular plant. So this plant has developed vascular tissue to transport water from down in here. I put it in here so it doesn't dry out because it's still very moisture and it has high moisture requirements. But from down here in the soil, it can transport water from there all the way to the tips here. So club mosses, they are vascular. And the other thing that kind of goes along with this evolutionary story is that not only are they vascular, they're now also sporophyte dominant. So their life cycle is sporophyte dominant. And the sporophyte here, it is green, and we were talking about how the gametophytes are always green. So in this case, the sporophyte is green, but the sporophyte is not fragile. So when I touch this, it's got some really good structure to it. Very fibrous almost. And then these leaves have a very thick coating of, it has a very thick cuticle, okay? So it's got a really nice, you can see how shiny. So this is not going to lose water very easily. These are spiky almost, like it's not painful, I guess, but spiky certainly, durable. So when we start thinking about sporophyte generations, we think about a much more durable structure. All right, and the other thing about club mosses is they make these very interesting reproductive structures. So the spores are produced from these strobili. And so the strobili are these reproductive structures that are produced at the very tips here. And I've got a couple of dried specimens, so let's take a look at those. So here's a dried club moss sprig, and I actually got this on a trail in Boone just the other day, and it's dried out, but it keeps its color really nicely. So this is a club moss, and you can see it still has that spiky texture there. It's got some little leaflets going on, which are kind of similar to this one. So similar structure. So they can look like mosses, but really train your eyes to see the difference there. All right, and then I also have some dried strobili, strobili, plural, strobilus for singular. And so you can imagine this sprouting from the tip of this structure here. So here's one, and here's a dried one. And you'll have all these like little, these just like little strands here. And all of this along here is a reproductively active tissue that is going to make sporangium, right? So sporangiums, and I forgot to mention this when we were talking about liverworts and mosses, the structure that produces spores through the process of meiosis is the sporangium, all right? The sporangium is the spore-producing structure of sporophytes. So they all start with the same couple of letters, which is always really helpful for me to keep track of what's going on here. The big deal here is that this structure that has the sporangium in it is going to produce spores, but the spores are going to be two different sizes, and those spores are going to associate sort of with male and female. All right, so the bigger spores are going to be megaspores, and they're going to be associated with the female generation of this life cycle. And then the male spores, the microspores, are much smaller, and they're going to be associated with the male portion of this life cycle. All right, that's called heterospory because the spores are different from one another. That's different than what we saw in liverworts and in mosses. So in liverworts and mosses, what we have is that they have homospory. So when we were talking about this little moss sprig, gametophyte, sporophyte capsule, the capsule undergoes meiosis to form spores, and all the spores will be the same size from moss. Same with a liverwort. When it produces that sporophyte generation that balloons out from underneath the fronds of the archegonial head, that sporophyte is going to produce spores that are all the same, and it's called homospory. So club mosses are just really interesting because they have all of these little tiny variations from the norm or from what we saw with liverworts and mosses. And so they're a really important evolutionary group. What's important evolutionarily about heterospory is that those male microspores are thought to be the transition point between spores and the formation of pollen, which we'll see in seed plants, okay? And then the megaspores, the larger spores, are thought to be the transition point between lower plants and seed plants in forming the ovule or the seed. All right, so this is a really important transitionary group, and I wanted to stress that. You don't need to know the life cycle, but these are really interesting plants, and they're very evolutionarily important. The last group that we're going to examine are the ferns, and so we will look at the fern life cycle. So let's take a look here. Got a couple of different fern samples. I want to start with a baby fern. So let's take a look at some baby ferns. All right. So here are some baby ferns, and I'm going to have to back up a little bit on this story because baby ferns don't just appear, right? They are formed through this life cycle process in a really interesting way. So let's talk first before I go through the life cycle. Ferns are vascular plants, so they're able to grow tall, and we'll look at some more mature versions that are much taller than this. This is a baby version, but ferns are vascular, and ferns are sporophyte generation dominant. So what you think of as a fern is actually the sporophyte generation. And we talk a little bit about how sporophytes are more durable. This is a maidenhair fern, so everything about it is delicate and fragile and amazing and beautiful, but that's not an indication of its generation. So sporophyte generation is a little bit more durable, and I'll say in comparison to the gametophyte generation. So let's start with how we know that the gametophyte generation starts for all plants. So we have a spore that lands. It's going to form a chain of cells. In this case, it would be a fern prothallus. The prothallus is going to continue to grow, and it's going to be kind of that abnormal shape. Let's do it a little bit more like this. And then as it grows, it's going to start getting a lot more like a heart shape, okay? And so big G for gametophyte generation of the fern. The gametophyte generation of the fern is actually still a very evolutionarily, it's reminiscent of an evolutionarily ancient plant. So it's going to actually have a lot of characteristics like a liverwort would. So it has a thallus. The thallus is very, very thin, very, very fragile. So fragile green gametophyte. And actually the gametophyte generation is going to still have rhizoids to kind of anchor it down into the soil. Okay, so very similar to a liverwort, but what's not similar to a liverwort is it's always a perfect heart shape. I promise, it's always a perfect heart shape, all right? And so the fern gametophyte is a very delicate structure. And as it's growing, once it gets to its mature phase, the mature phase is going to still be really small. All right, and so I actually have a fern gametophyte that I'd like to give you a quick look at. Here's our fern gametophyte, and you can't even see it, so I'm gonna have to zoom in. It's so small. All right, but I got these from some spores of ferns that I propagated, and if you would like more information on how to grow your own ferns, I'd be happy to talk to you. It's really easy. You just microwave some soil and then sprinkle some fern spores on there, and we'll. But I would be happy to talk about it more with you if you would like. Just get in touch. But here's our fern gametophyte. So it requires a lot of water. You see that I have a lot of water in here, and the gametophytes are very, very fragile, and they stay super, super small. And you can see this one kind of has that heart-shaped structure here. And so when we are looking at the fern gametophyte, we have our heart-shaped structure. And when it's mature, it's still going to be super small, but it's going to form these structures. So it's going to form some structures on kind of the outside edges of the leaves. And then it's going to form some other structures kind of in the crook of this heart shape, okay? These are going to be the archegonia. So A-R-C-H-E, you can guess what that is. That means that these are the female structures. And then the antheridia are going to be on the edges here of the thallus, kind of in the lumps of the heart shape. And so anther, we know, is indicative of the male parts. Technically these structures can have only female or only male parts, or they can have male and female. I wouldn't worry too much about that. The important thing is that as we've seen before, the male parts produce sperm, the female part is going to produce an egg, and when sperm meets egg, we have a zygote that forms, okay? So once sperm meets egg, we're gonna have, just gonna put a big dot in the middle, and that's gonna represent our zygote. So the gametophyte generation with 1N sperm going to 1N egg, we now have a 2N zygote that has formed here. And the 2N zygote is going to produce more cells through the process of mitosis, and it's going to form our sporophyte generation. And at first, it's really small, but it's going to be our fern frond. So I want to draw it like this. Okay, fern frond that is growing directly out of this heart-shaped gametophyte stage, all right? And so if we were looking at this, the gametophyte stage is down here somewhere, but it's so small. And even at this really small fern stage, the gametophyte has already sort of been absorbed. I mean, it's not actually absorbed, but it kind of withers away and dies because this structure is growing directly on top of it. So the sporophyte is growing directly on top of it here. And so the sporophyte will continue to grow out of that gametophyte tissue until the gametophyte tissue just withers and dies, and then it will continue to grow. So, and I know that these are immature sporophyte phases because they're still kind of fragile to the touch, still kind of fragile. But if I pulled this out, this sporophyte phase would be vascular and it would have roots down here, right? So one thing that happens that I want to show on this picture is those rhizoids are still here, but as the sporophyte grows, it's going to grow up and down. So the sporophyte frond, what we think of as a regular fern frond, is going to grow up, and then the roots are going to start growing down, all right? And so this structure is going to be anchored to the ground with roots, or initially with the gametophyte rhizoids, but then the roots are going to grow from the sporophyte, and it's going to be a vascular plant here. So just something to think about there. All right, so we looked at a couple of immature versions of our sporophyte generation. Let's look at a mature form. Move out of the way, drawing. Okay, so here we're smaller and then bigger, but I want to show you the underside of the frond. The frond is, there's nothing special about the underside of the frond, just green, leafy, nice, all right? And then we're going to get even bigger. All right, and this is a mature sporophyte, and I know that because on the underside of the frond, I can see these brown structures, all right? And the brown structures are indicative of something, so I'm going to just zoom in so you can really see it. All right, you can see these brown lines. All right, and that's different than when we looked at this structure and we looked at the underside, and there's not really anything on the underside of these fronds. Okay, so immature sporophyte. Mature sporophyte. All right, and so what are those brown structures? Well, those are actually, the structure itself is called a sori. Sorry, you keep getting little snippets of like my shoulder or my head, my ponytail. It's working in a limited space here. I really appreciate your patience. So when we have our mature sporophyte generation, what we're going to do is we'll draw our frond. So here's our mature fern frond. Man, I'm having luck with these pens. All right, here we go. So we've got our fern frond here. And so when it's immature, the bottoms are going to just be like leaf. But once it matures, it's going to produce these sori. So in some ferns, the sori are circular. That's probably what you're more familiar with. And I can show you a version like that. All sorts of versions. So here in this version, you can see that the sori are circles. Okay, and the one that we were just looking at is not circles. So let me take a little cutting off of here. This looks, the one that we were just looking at, that one had kind of the lines. Okay, this one's like polka dots. Had lots of little tiny sori. Okay, and so what are sori? So sori are collections of sporangia, right? So this is the sporophyte, and eventually it's going to need to make spores, but it does so on the underside of the frond. And so the sori is a collection of lots of sporangia. And so the sporangia kind of look like this. They've got this like funky little curl and a backbone structure. Okay, so here, imagine this is kind of looks like a backbone. And then the spores form inside of the sporangium. Okay, and the sporangium, there's like lots of them all together, all right? And so let's just keep drawing them. And when they're all clustered together like that, they're gonna look like a spot on our fern frond, all right? So sori, S-O-R-I, is a collection of sporangium, or sporangia for plural, excuse me. So single sporangium, a collection of sporangia. And then each sporangia with its backbone structure here is going to produce spores. All right, and so then eventually the spores will disperse out into the environment. All right. So sporangia in a cluster make a sori. So 1 sori, 2 sori, 3 sori. Let's take a look at this if we can underneath the stereoscope. All right, and so here we can see each individual sori. It's hard to do this. 1, 2, 3, 4 sori. All right, and each of those is a cluster of sporangium. So I want to also take a look at a sample of sporangia so that you can see what they look like. Give me just 1 second to find the slide. All right, and so the sporangia, this is what we were talking about, those curled structures. So let's find a really good one. Ooh, I like this one. There's a couple here that are good. Okay, so we'll do this one right in the middle, and let's go to 10x. All right. Yeah, okay, so if you're looking here, I'm not sure that in the recording that you're going to be able to see my mouse, but this top part that looks like a stem, like a little stem part here, that's where the sporangia, the sporangium, because it's a singular, excuse me, is going to connect to the fern frond. And then there's going to be lots of these. This is a sporangium, lots of these in a cluster to make that sori, or that circle on the underside of the frond that you saw. This is microscopic, so a single one of these wouldn't make a dot on that fern frond. So think about it that way. There's going to be lots of these in a cluster in order to make that sori stand out on the bottom of the fern frond. But right now we're just looking at a single one. So this is the entire sporangium. And then here inside, the sporangium makes the spores, and this is all on the sporophyte generation. All right. So once these spores disperse into the environment, they're going to germinate and then make that prothallus and then grow into the fern gametophyte. So we have successfully come back to the beginning of our fern life cycle. All right, y'all did so good. Thank you so much for sticking with me here. We looked at some specimen, and we didn't look at as much of the microscopic specimen for these. So there is, if you go to As You Learn and you look at the activity resource, there's multiple activity resources. So there's one for liverworts, there's one for mosses, there's one for ferns. And when we look, and for phloemosses as well. And when you look at those activity resources, a lot of what that's going to be is some of the specimen that we looked at in lab, examples of, gametophyte versus sporophyte generations, but you're also going to see a lot of microscopic specimen. And so that's one thing that I can't do here is look at some of these specimen because with plants, the specimen is like a little bit, the microscopic specimen is a little bit too big for a compound microscope, but not quite big enough to look at underneath the stereoscope, okay? And so it just doesn't translate very well, but you do have the activity resource, and it'll give you all of the microscopic specimens that you need to look at. So some of the things you might want to look at in the microscopic specimen of that fern gametophyte. We just looked at the fern's sporangium. sporangia. You might want to look at the liverwort sporophyte. You can actually see the spores forming in the sporophyte. So just a little hint tip that looking at that activity resource for each of the different subgroups is really important when you're studying for the lab practical, and then reviewing this video and the specimen. So how can you identify a liverwort in real time? Like if somebody just passed you a liverwort, how would you identify it? Well, you'd have to know it's not a fern. Well, a fern has that frond shape, whereas a liverwort has the liver shape. So really, when you're studying for the lab practical, I want you to look at the specimen and say, how can I identify this in comparison to other plant specimen? All right. If you have any questions or concerns, please get in touch with your lab instructor or myself, Cara Schreiber. I'm the lab supervisor. And otherwise, just I hope that you enjoy plants, because I think they're really different than us, but they have a lot of really cool features. And I'm glad that I was able to walk you through seedless plants this week. Your lab instructor will walk you through seed plants at the end of the this upcoming week. So thank you. Y'all have a good day, and until next time. Thanks.